last video I kind of talked about real basic stuff, why do we need statistics, and some kind of basic terms. The other thing I need to talk about before we kind of dive into what tests should you be doing is to think about different statistical philosophies. And there's a few different versions of these. And I want you to be familiar with them in case you're reading a paper and you see something and you have no idea what that means. And that'll probably happen anyway because there's so many specialized kinds of statistics that eventually you're going to read a paper and they're going to talk about some methods that you don't know anything about. I read papers and they talk about methods and I have no idea what they're talking about. And I've been doing statistics for a very long time. So that just goes to show you that there are a lot of different kinds, but a lot of them are based in a few different philosophies. And so I want to point that out. There's one type where you use prior information. So this graph in the right-hand corner kind of illustrates that, where you have some prior beliefs. So maybe you collected some preliminary data, or maybe there's another paper that tells you something about the question you want to ask. So your prior beliefs are here, and then you collect data, and that's your evidence. And then what you end up with at the end is what they call posterior beliefs. So basically, they use the prior information and the data you collected, and they come up with the answer that's in between. Bayesian statistics, they're called Bayesian statistics because they were invented by a theorem from Thomas Bayes. They're more computationally intensive. So a long time ago, like before I was born, in the 50s and 60s, almost nobody did Bayesian statistics because it would take you forever to do without a really good computer. These days, in the 2000s, Bayesian statistics have become much more prevalent than they were in the past because we have computers that can do it really quickly and you don't have to calculate anything by hand. Some criticisms are that using prior beliefs, you basically can specify how much weight those prior beliefs or previous information is taken into an account. You can use something called a non-informative prior, which means that basically the prior beliefs don't actually matter in the calculations. But this is some criticisms of this particular philosophy. Other types of statistics often use no prior information. So you collect your data and you answer your question entirely on the information that you collected in this study. People that use Bayesian statistics will criticize this because if you've been doing research on this subject for a long time, why would you start completely from scratch? So that's one criticism. But, priors, the subjectiveness that can influence the answer you get is a criticism of Bayesian. So it depends on what you're doing and what your philosophy is, which type you will use. In a standard statistics class, you will learn frequentist statistics, which don't use prior information. So those are not Bayesian statistics. Frequentist statistics, that's what has a p-value, which we'll talk about in a minute. The computations are much simpler, which is probably why you learn them in a basic statistics class, is you can calculate out and do it, but it's a little bit harder to interpret. So frequentist statistics, I think, is what you'll still find to be the most common thing, is the most commonly taught, and it is what you'll see in papers a lot. And pretty much anytime you see a p-value, that's a frequentist statistic. The data needs to come from repeatable random samples. The parameters that we're trying to estimate are fixed. So usually you're trying to estimate the average of something or the difference in the average between different groups. And we use what's called hypothesis testing in frequentist statistics. And you compare two different hypotheses. This is not the hypothesis you necessarily make about what you're going to find in your experiment, you have a null hypothesis, that's that there's no pattern. And so if you're looking at different groups, for example, we can go back to our crabs and whether they're different sizes for different species. The null hypothesis would predict that there's no pattern. So that would say that the difference between the groups are no greater than we would expect from random variation. So that means that essentially, if you look at all the variability among crabs, in general, there's going to be no difference between the two species that we're looking at. And if we find this to be the case, we often say that we failed to reject the null hypothesis, which I know is extremely convoluted, but that's what technically is correct if you learn it from a statistician to say. Now, I don't usually see that written in a paper, so. The other hypothesis is the alternative hypothesis, and usually this is that there is a pattern. So your alternative hypothesis may be that the two species are different in mass. And you can also make ones that are in a particular direction. So you might predict that species A will be bigger than species B. That would also be an alternative hypothesis. And this is saying that the differences between the groups are greater than we would expect from random variation. So if we looked at just the variability among crabs altogether, we're going to see more differences between the two groups than just among individuals within the same group. And if we find that this is the most likely scenario based on the statistics we do, we would say we reject the null hypothesis. So there's enough differences to say that we can reject this idea that there's no pattern among our groups. So that brings us to p-values. It's the probability that the observed or more extreme differences would be observed if the null hypothesis were true. That is really convoluted. It's basically the idea that if we took an infinite number of samples of the same size, so we want to know if the difference between these two species of crabs that live in the marsh outside of Lumcon, if those two species are different. But we only took a sample of 10 crabs of each species. It's asking what the probability that we'd get the difference that we see in our sample if we took an infinite number of different samples from Lumcon. If we had endless amounts of time to sample all the crabs over and over and over again, we looked at 10 crabs of different sizes and we got all different individuals. You'd get different averages if you grab different crabs. And the question is, what's the probability that what we got in our sample, that we would have gotten that or a more extreme difference if we knew that there really was no difference between the two species of crabs? Now, I told you that frequentist statistics are much harder to interpret. So what I just said was very convoluted and confusing. That's a hallmark of frequentist statistics. But a lot of times when we write it in a paper, we would just say that the two species of crabs were significantly different, which is much easier to understand. But to be technically correct, you have to say something very, very complicated if you're a statistician and you use frequentist statistics, which is the p-values. P-values, if we want to interpret them, so we do a statistical method, we get a p-value. Smaller p-values mean that it's less likely that the data matches the null hypothesis. So basically, if the p-value is smaller, it's more likely that the two groups are different. And if the p-value is really big, it's more likely that the null hypothesis is correct and that there are no difference between our groups. Now, p-values can vary between 0 and 1. They're probabilities. If you get a p-value of 1, that means there's pretty much definitely no difference between your two groups. That's a really big p-value. And usually if we call something significant, that's when the p-value is 0.05 or smaller. So a really small number. And that basically means that we're willing to accept a 1 in 20 chance or a 5% chance that we're wrong. And so we'll say that there's a difference if there's only a 5% chance that there's not. So basically, in all practical purposes, you want to look for a p-value of 0.05 or smaller, and that will mean that your groups are probably different from each other. And if your p-value is greater than 0.05, you will probably say that there is no difference between your two groups. Or you can say that you failed to reject the null hypothesis. So again, as I said, significance, we usually use 0.05 as our threshold. And I do want to point out that statistical significance is not the same as biological significance. So if our two species of crabs, if they're about a kilogram, but our statistical differences are only a milligram, is that going to have a real functional difference? They might be statistically different in terms of only a milligram difference, but functionally in the environment, maybe a milligram isn't enough to change how much food crabs eat or how they affect other organisms or things like that. That would not be statistical significance without biological significance. Similarly, maybe you have a very small sample size and you observed a really big difference, but you were only able to measure a few things. This happens a lot when we do large ecosystem scale experiments. You might have 2 large plots of marsh that got oil on them from the BP oil spill and 2 large tracts of marsh that didn't get oil on them. That's a really small sample size. You might not be able to get statistical significance, but if you see large differences between the oiled marshes and the not oiled marshes, then there might be biological significance without statistical significance. And a lot of times when you have biological significance and not statistical significance, there is a problem with sample size. So you might be trying to measure something and it's just not possible to get a lot of samples. There might not be a lot of marshes to compare, or it's completely impractical to measure a lot of marshes. So that's my talk on p-values. So I'll give you an example, and we talked about this kind of already. Are males bigger than females? The test shows no difference, so the p-value is greater than 0.05, and that means that we failed to reject the null hypothesis. And so that means that males are not bigger than females. They're roughly the same size. There's variation among individuals, but it's not due to sex. Another important thing to note is that the absence of evidence is not evidence of absence. So failing to reject the null hypothesis is not accepting the null. The reason for that is that you have a 95% chance that you're going to reject the null hypothesis. So a lot of times it may be that the difference isn't very large. It might be that there's a lot of variability among individuals. So the biggest individuals might always be males, but there's so much variability because you have lots of individuals that are different ages and have been growing for different amounts of times that there might not be a statistical difference between males and females. You might not have a big enough sample size like we talked about. You're more likely to get a statistically significant effect if you have bigger sample sizes. If there is a true effect, you're more likely to detect it with a bigger sample size. And so these are some things to think about. So just because you don't find a difference when you do a study, it doesn't mean that there's not a difference. It just means that you don't have the evidence to support that there is a difference. You might need to do another study that's more robust, or you might need to control for other variables.